Our next speaker is Hilary McGraw, who's going to tell us about Wnt signaling and collective cell migration.
Hello, thank you for this opportunity to speak. Today, I'm going to be telling you about the role of Wnt signaling in organ development. So I'm in particular interested in a particular aspect of cell migration, collective cell migration, in which, let me see if I can find the precursor, in which cohesive cohorts of cells will migrate together as units. And often within these units, there will be cellular specialization in which there are leading cells which tend to send out filopodia and explore the environment and direct migration, while directing pools of cells behind them that are often more differentiated. This process is very important for organ formation in many aspects of embryonic development, including Cooper's vesicle, like we heard about earlier, vascular sprouting, and neural crest migration. And in addition, in a more developed organisms, collective cell migration is often an aspect of wound healing. And so it's very important to understand the mechanisms of collective cell migration, both as an aspect of developmental biology, but also many of the same mechanisms are misappropriated in some invasive cancers. So I am using development of the zebrafish lateral line as a model to study collective cell migration during embryonic development. And what I'm showing you here is a 2-day-old zebrafish embryo labeled with the transgene claudin B, which very nicely marks the newly developed lateral line, a series of mechanosensory organs arrayed along the trunk of the embryo. Usually by 2 days post-fertilization, there are approximately 5 or 6 of these mechanosensory neuromasts, as well as a cluster at the end of the tail called the terminal cluster. The lateral line develops very nicely through the migration of the lateral line primordium, this cohort of approximately 100 cells that migrates along the trunk of the embryo between 24 and 48 hours of development, depositing these mechanosensory neuromasts as it goes. So when we focus in on the lateral line primordium itself, what we see is that this collection of cells contains specialized structures with these highly protrusive tip cells that direct migration, proliferating progenitor cells that are not yet polarized at this stage, as well as more polarized epithelial cells, which form these very nice rosette structures that will mature and be deposited as these mechanosensory neuromasts. So what we want to know are what are the cellular and molecular mechanisms that regulate collective cell migration in the system. And to do that, one mechanism we're using to study that is an ongoing forward genetic screen to identify mutants that show defective lateral line formation. And what I'm showing you here is one of these mutants I pulled out of the screen. So this top panel is a wild-type embryo showing the full extension of the posterior lateral line system, this terminal cluster at the end of the tail, and a mutant embryo, LEF1, in which we see loss of this terminal cluster set of neuromasts, as well as premature stalling of the primordium and loss of primordium organization. And so, as I'm sure you've already guessed, this mutant is in the Wnt transcriptional activator LEF1, which acts in conjunction with nuclear beta-catenin to drive Wnt-mediated gene expression. And when we lose this LEF1 protein, we lose most aspects of Wnt-mediated gene activation. And this is critical because our lab and several others have shown that the Wnt signaling pathway is critical for maintaining progenitor cells within this leading region. And it's just a brief bit of work that I did to demonstrate this. I used Kaede photoconversion to label these progenitor cells at the onset of primordium migration in these wild-type embryos, shown here, these converted cells in red, in both wild-type and mutant embryos. And at the end of migration at 48 hours, I see that in wild type, these progenitor cells have proliferated and formed the neuromasts of the terminal cluster. While in contrast, in LEF1 mutant embryos, these cells, although they remain proliferative, have exited the progenitor zone and have been prematurely incorporated into neuromasts, leading to a loss of progenitor cells within the progenitor region, and effectively leading to loss of proper collective cell migration in the lateral line. And this is quantified here, just showing that although these cells remain similarly proliferative by 48 hours, we see a difference in where they're deposited within the lateral line, developing lateral line system, indicating that loss of Wnt activity through LEF1 leads to a loss of proper progenitor cell identity in wild type and mutant embryos versus wild type embryos, where they actually stay within the progenitor region and form terminal cluster neuromasts. So we next wanted to ask, we know now some of the molecular regulation that is required for proper collective cell migration in this organ system. What are the targets? What are the actual mechanisms that are regulating this process? So to do this, I undertook a RNA sequencing screen to compare wild-type embryos to LEF1 mutants, as well as embryos in which Wnt signaling had been overactivated using the pharmacological chemical bio, which leads to loss of the destruction complex and accumulation of beta-catenin and Wnt activation. In the absence of a good specific marker for the lateral line primordium, I manually dissected out primordia from collections of embryos and performed RNA sequencing on these isolated primordia. And so what I'm showing you here is just a heat map showing the normalized log base 2 changes of gene expression in our wild type LEF1 mutant in biotreated conditions. And in particular, I'm interested in looking at factors that are either downregulated in these LEF1 mutants compared to wild type or Wnt overexpression, or conversely upregulated in these mutants compared to wild type and biotreated. And when I look at what I pulled out of the screen, many of the factors are involved in embryonic development and in particular in cellular behaviors such as motility, cell cycle, and cell death, which would be potentially interesting targets for looking at the mechanisms that regulate collective cell migration, cellular behavior within collective cell migration. And today I'm just going to tell you about one of these genes we've looked at, FAT1B, and I would also like to thank the Dorsky Lab, who, while we were conducting our screen, shared their gene list with us as well, and we also found this factor in both RNA sequencing screens. So on the stop panel, I'm showing you an in situ for LEF1, a known activator and target of Wnt activity that is nicely expressed in the leading region progenitor cells within this primordium. It's downregulated in LEF1 mutants and upregulated, its expression is upregulated following Wnt overactivation. So one of the genes we identified in our screen, FAT1B, is nicely expressed throughout the lateral line primordium in a wild type embryo, downregulated, or at least partially downregulated in LEF mutants, as you can see, is also upregulated when Wnt signaling is overactivated, suggesting it is in fact a Wnt target within the lateral line primordium. So FAT1 was initially identified in Drosophila, but has also been found in vertebrate systems as well. These are atypical cadherins. They're very large proteins, and in the zebrafish, this is almost 4,500 amino acids. They contain a very long extracellular domain containing, in the zebrafish, approximately 34 cadherin repeats, a laminin EGF-like domain, a transmembrane domain, and a cytoplasmic domain, which in FAT1 in a mouse system has been shown to interact with various aspects of the actin cytoskeleton, possibly regulates beta-catenin, cytoplasmic beta-catenin, as well as interacting with the Hippo pathway. So there are several different functions that FAT1 may be playing in maintaining cellular mechanics during collective cell migration. Work specifically in mouse has shown that FAT1 regulates nervous system formation, neural tube closure, eye and kidney development, and the mouse knockouts die just postnatally due to improper kidney formation. Specifically, FAT1s have been shown to be expressed in proliferating cells and are downregulated once cells exit the cell cycle. And as I said, it also may regulate Wnt signaling by acting on cytoplasmic beta-catenin, perhaps sequestering it away from the nucleus and leading to downregulation of Wnt signaling. And mutations in FAT1 have been associated with many different invasive cancers, suggesting it may also be an interesting disease target. So I generated CRISPR-Cas mutant and the FAT1B in Zebrafish, which nicely shows a truncated lateral line. So once again, I'm showing you a wild-type embryo on the top panel with a fully formed posterior lateral line. And in FAT1B mutants, we see once again this loss of the terminal cluster, premature stalling of primordium migration, and an anterior shift in where neuromasts are deposited, suggesting that FAT1B is in fact involved in collective cell migration. And when we look at movies of FAT1B mutants, we can see that primordium migration is much slower than in wild type, and as well we also see what looks like perhaps inappropriate cell-cell interactions. You can see these appear to be much more condensed compared to a wild type lateral line primordium. And I initially looked at how cells are behaving within the progenitor region in these FAT1B mutants, once again using Kaede photoconversion, where we see in wild type, we very nicely see that these leading progenitor cells form the terminal cluster neuromasts. In contrast, in FAT1B mutants, we see that these labeled cells are incorporated into neuromasts, but are often completely excluded from this more leading region that forms the terminal cluster. And when I quantify that, I show that I am able to convert initially the same number of cells at the onset of migration. These cells appear to be normally proliferative when I compare mutant cells to wild type. But when I look at where they're actually deposited, well, wild type cells form primarily these terminal cluster neuromasts. In FAT1B mutants, these cells are actually shifted anteriorly, once again suggesting perhaps a similar mechanism to loss of LEF1, where FAT1B acts to maintain these progenitor cells in their proper region so that they can form the complete lateral line. In addition, in zebrafish, as is a common problem, the FAT1 gene was duplicated, so there's also a FAT1A in fish, which is expressed within the lateral line primordium. May not be Wnt sensitive, although I do see some upregulation following biotreatment, but may play a compensatory role for loss of FAT1B. So it would be very interesting. I'm in the process of knocking out FAT1A to see what a double mutant looks like. So for my future work, I would like to know specifically what is the role of FAT1B in mediating cellular behavior during collective cell migration. Is it is it causing cell-cell interactions, altering Wnt signaling, altering the cytoskeleton during a cellular migration, or interacting specifically with the Wnt signaling pathway? And does FAT1A play a role in lateral line development and collective cell migration? And in the future, I will continue culling through this list of RNA sequence of target genes that I've identified to see if I can find a mechanism that's regulating collective cell migration. And with that, I would like to thank the Nachaporic Lab and our collaborators, the Dorsky Lab and the Nicholson Lab, for a lot of bioinformatic help and your attention. Thank you.
Do you know if FAT1B is the only functional target downstream of LEF1? Have you tried to rescue the LEF1 phenotype by injecting the FAT1B?
I haven't. Because FAT1B is such a very large protein, we're actually having some difficulty figuring out how to generate a good construct for rescue. I have acquired some of the mouse constructs that have been generated in other contexts, so we're in the process of seeing if we can rescue. And I also don't know what a double mutant looks like. I think it would be interesting to look at that.
So, very nice talk. Struck with the sort of partial nature of all your mutants. They make the lateral line, but they just kind of stop early. Is that a maternal compensation, or what's going on?
For LEF1, it is not. We are actually able to generate homozygous mutants and get progeny from those. So it doesn't look like there's a maternal contribution for LEF1. It could be that there are some signaling pathways that are setting up the initial structure of the lateral line primordium that are outside of the Wnt signaling pathway. Yeah. We don't know what that is yet, though.
Hi, so if I understood correctly, the phenotype you're suggesting is that the cells that are supposed to proliferate at the front get deposited too soon, and then the lateral line basically runs out of juice before it gets to the end. But at least in the FAT mutant, it seemed like the number of clusters was not less there, even maybe were more of them. They were just more closely packed together, so it seems maybe...
So there could very well be some aspect that's both proliferation and cellular adhesion, keeping them within the context of the migrating primordium. So I don't know the exact mechanisms that's contributing to cell number, but you're right. I think there could be some other mechanism that's regulating both proliferation and deposition.
Could there be any difference in the apical basal polarity of these cells?
I don't know that yet, but that's definitely something I'm looking at, is how the cells are structured within the context of the primordium. Thank you.